Good afternoon. Welcome on the second lecture on machine learning in bioinformatics. Today I will complete the introduction. Actually, I have just one last slide to the part, and then I will review some notions and notation, and then we will start with the topic of unsupervised learning. So this slide motivates technology, which is old, 27 years, which enables to detect DNA. And actually, it enables to detect some short strings of DNA, whether they are present in sample or not. And these are called DNA chips because they enable to detect several thousands of such fragments of DNA in one experiment. And the experiment consists in the following. There are spots on the chip. For the size of the chip, you can see the picture top right corner. And each spot on the chip contains short pieces of DNA, one-stranded DNA, of length several tens, let's say 20 to 30 or 40, mostly at most 40 bases. And if on each spot of the DNA chip, we have the same short piece of DNA. It's on the picture on the left. I would like to use at least highlighter. So these are the pieces of DNA, and if we have some sample, and in the sample there is a complementary DNA string, it can bind to this short DNA string, and the bound is strong if there is a complementary part, even in some long DNA string. And if we mark the samples by some fluorescent color, then we can observe this color after the experiment. We inspect the chip under microscope, and we check the color. On the right, on the big picture, we can see experiment where we have two samples, normal and some sample from a tumor or possible tumor, and the DNA from the normal sample is marked by green fluorescent color and tumor by some magenta color. And then if we put together these both samples mixed on the chip, on the spots where they found complementary substrings in the DNA, and they will bound to the spot. And under microscope, we can check the color of the spot. The color is after lighting some laser on the chip, and if the spot is green, then we know there was this substring corresponding to the spot is present in the normal sample, green. If it is if the color and not in the tumor, in the sample from tumor. If there is only, if the substring is present only in the DNA from tumor, then the color will be magenta. And if the sample is present in both normal and tumor samples, then the color will be yellow. So in this way, we can detect whether some substring is in DNA. And what DNA? It's usually used for detecting whether some gene is active. Active genes means that it's copied from DNA to message RNA and then used for the inside the body of the cell. And if we know that there is some gene of interest, the gene can be long, and then we take several substrings, maybe overlapping from the gene. Such substrings are used as the sample on the spots on the DNA chip. And we are interested whether the gene is active. So we extract the DNA from living cell, then mark it by a fluorescent color, and then apply it on a DNA chip, on microarray. And if the gene is active, then it should bind to all these spots corresponding to substrings from the gene. So this is the technology for detecting substring. As I said, the number of spots on such chip is several tens of thousands. So it is possible to detect many different fragments of DNA, and it's used for various experiments. So there are special microarrays designed for humans, for mice, and for other organisms. Okay, now let us continue with some statistics. Very little statistics. Actually, we will work throughout the lecture. We will work with some variables considered as random variables, and for describing random variables, we need some notions from statistics. I suppose that you already know all these notions, just to be sure that you understand it, and just to introduce the notation, so you could follow my further slides. So if we have a random variable, and a random variable is from discrete probability distribution, it means that there is a discrete set of values which can be of the value of the random variable. Then we can describe the distribution how often we can encounter given value for the random variable. So this is the probability distribution. And in case of discrete probability distribution, it's just enough if we know the probability for each possible value. So we use probability mass function, and P of xi is one observation, one value, is the probability that the random variable x has the value x sub I. And of course, if we sum for all the possible values, of the random variable, if we sum the probabilities, we should obtain probability 1. For continuous variables, so where the variable can be, for example, real value from some interval, then of course the probability for single value is how much? Usually 0. Usually 0. It need not be that, but it's usually 0. So for describing continuous random variable, we use, instead of probability mass function, we use density function. And density function is such function that the probability that the random variable is from interval AB. So the probability that x is at least a and at most b is the value of integral between a and b of the density function f of x dx. And also sometimes we also use cumulative density function. Cumulative density function, which is the value of the integral from -infinity to x. So actually, it's this formula for a equal -infinity. So this is the probability that the value of the random variable x is at most x. And of course, if we compute integral from -infinity to +infinity of the density function, we must obtain 1. Is there any further requirements for the function, or can we say at least some restriction for the values of the density function? Can the density function have an arbitrary value, arbitrary real value? Or should it be from interval 0, 1? What do you think? Who thinks that the density function can have any value, any real value? 1, 2. Oh, this is wrong. There is some restriction. Who thinks that it cannot be higher than 1? You are right, it could be higher than 1. And can it be 0? Can the density function has value 0? Yes. Can it be negative? Why not? Actually, if we go deeper in the definition of density function, we would find that there are some more requirements, but what is most important, we have this formula for computing probability that the value of the random variable is from given interval. This one. And the probability must be non-negative. From a mathematical analysis, if the function has, for example, some singular value for one value, it will be -1, and all remaining values will be non-negative. Then the integral still would be non-negative. But in general, it must be non-negative. But it can be any positive value can have, and the only restriction is that the integral from -infinity to +infinity of the density function must be 1, or the area under the curve must be equal 1. Okay. Expected value. So if we have some numeric random variable, then the expected value of the random variable is some value to which tends the values of the random variable. But the random variable need not to be constant; it can change. So the expected value is defined as the sum of, in case of discrete random variable, value of x times probability that the random variable capital X has the value lowercase x. Or in the case of continuous random variable, it's integral of x times px. What is px? The density function. x times dx, and this is the expected value. If we have some samples, so we can measure the random variable, and we have capital N measurements, then we can estimate the value for the expected value, and the estimate is mean. So mu equal sum of the values, sum of xi divided by capital N. Capital N is the number of samples. It can be shown as this is so-called unbiased estimate. What is the meaning of the unbiased? What does it mean that it is an unbiased estimate?
Approaching infinity, we get really the beta. That's we are or we are
no, this is not the proper n equal n going to infinity. It's not necessary. If we the x is random variable, so every measurement could return different value. If we compute average, so the sample average, then we obtain value which is also not constant, variable. Actually, this is a random variable, and that the expected value for the random variable for this expected value for sample average must be the expected value for the random variable x in this case. So even for fixed capital N, we can perform many experiments. In each experiment, we take capital N measurements and compute average. And this value is actually a random variable. And the estimate is unbiased if this variable has the same expected value as the original random variable. I'm thinking about that because we will look at in the next slide at variance, and there is a difference. So expected value for the sample average equals to the expected value of the random variable, even without n going to infinity. Therefore, unbiased estimate. Variance is a measure how widely the value of the random variable differs from the expected value. And it's measured as a expected value for the square difference. So if x is the random variable, we compute x minus expected value of x, and this value square, then expected value for the square difference is the variance. And of course, we can compute with expected values, and here we have expected value of some expression which is square of some smaller expression. So if we rewrite the expression, this will be x squared minus 2x times expected value of x plus expected value of x squared. But now we will compute expected value. So expected value of x squared is still expected value of x squared. We can split it linearly. From the second term, we will have expected value of 2 times x times expected value of x. Then expected value of x we can put in front of the expected value, and we obtain that it is actually 2 times expected value of x times expected value of x. And from the last term, we have still expected value of x squared. But this term can be subtracted from the last one, and we obtain that it is expected value of x squared minus square of the expected value. Okay, this is theoretical. And if we have a lower variance means that the values of the random variable are in general closer to the mean, and higher variance, they are spread farther. So these are density functions for some normal distribution with a lower variance and with higher variance. And we would like to estimate the variance. Sometimes you can see the following formula that we have, as in previous case, we have capital N measurements, and we can compute the square differences between the value of the measurement and the expected value. We don't have the expected value. For the expected value, we can use just the estimate, the sample mean. Okay, so if we use the sample mean mu for the sample variance, then this formula is not correct because it underestimates the true variance of some factor n-1 divided by n. So the sample variance, so the correct formula for sample variance, which gives us unbiased estimate for the variance, is the sum of square differences divided by n-1. Of course, if n is big, capital N is big, then the difference between these two values is very small. But for not so big, capital N, this could make a difference. Interestingly, this formula above, first one, is based on so-called maximum likelihood estimate, and the second is for obtaining unbiased estimate. Both estimates are almost the same for big values, capital N. But be aware of that usually if we compute sample variance, then we should use this value, as this is better approximation for the variance. Now this is extremely important. Throughout whole lecture, I will use this notation introduced in the following two or three slides. And you should remember that, otherwise you could not follow properly. I will try to recall the notation for you several times during the lecture and following lectures, but nevertheless be aware of that. So we have some observations, and these observations are described by values which take some attributes which take some values. All the observations we will store usually in metrics of capital X, because most of the methods we will cover in the lecture will work with numerical variables. So we will describe usually the objects using some values of some attributes. So we will always use capital X as the matrix containing all the observations, where one row of the matrix is one observation. So if it is some ith row, this will be xi1, xi2, and so on, xid. D is the number of attributes. And altogether, we will have capital N observations. So the top left corner will be value for the first attribute for the first observation, 1, 1. And left bottom corner will be the value for the first attribute of the nth observation, n1. And of course, right bottom corner will be value of the last attribute for the nth observation, so nd. And of course, here it will be 1d. So this will be the observations. When we will compute with that during labs, we will usually have this table representing this matrix. So each row of the matrix will correspond to one observation, and each column will correspond to a value for one attribute in all observations. And the notation is, if I use a lowercase italics x, this will denote, and if it has a vector of length d is the number of attributes. This corresponds to one observation. However, usually in algebra, when we speak about vectors, then we usually think by default that this is a column vector. So xi is one row, is the ith row of the matrix capital X. But if I take it as a vector, one row, then it's column. So therefore the notation that it's vector xi1 till xid transposed, because it is a column vector. Okay. If I use lowercase letter, but boldface and without italics, this will denote one column of the observation matrix X. So boldface X here is the J's column of the observation matrix, and it's the value of the J's attribute in all observations. So we can write that the capital X observation matrix is a concatenation of these vectors boldface x1 till boldface xd. These are column vectors, and put together they will form the matrix X, capital X. And the same matrix can be written as x1 transpose, because if I write x1, it's a column vector, but we need x1 as one row of the table, therefore transposition. So this is this representation, and it can represent the observation matrix capital X. If we have a target variable, then y sub I will be the ith observation, the target of the ith observation, and the whole observation table consists of pairs. x1 is the observation described by the attributes, and y1 is the target variable. Of course, in general, target variable can be also a vector, but in most cases it will be just one number. Therefore, this is, I think, the last slide of this part. If I write a scalar value, a from the set of all real numbers, then I will write it as in lowercase and italics. If it is lowercase and bold, and it's usually from a vector of length capital N. It corresponds to the column of the observation matrix. If it is vector, but of length different than N, then we will write it in lowercase and italics letter. If it is matrix, we will usually write it in capital and bold, without italics. And if it is a random variable, it's usually capital and italics. So capital, uppercase, and italics. I will try to prefer to keep it in this form. Yes, no. Okay. Any questions? So this is the end of this part. Let us switch to the next. Unsupervised learning. So for sources, I have two sources for this part. One is the Young book, Machine Learning Approaches to Bioinformatics, accessible through our library and online, complete text. And the Bishop's book, Pattern Recognition and Machine Learning, which is also available complete text online from this link. Let us start with an example. We study some plants, and the plants, when the plants are growing, they generate many thousands of metabolites. Metabolites are some intermediate products during metabolism. And usually it's used for small molecules. You can see that if definition at most 900 Daltons. So the weight of the molecule should be not higher than 900 weights of a proton or neutron. And we can use mass spectrometry to detect mass of metabolites. What is mass spectrometry?
It's difficult to explain that. Basically, split the molecule really fast, then ionize it, shoot it with electrons, measure its weight.
Yes, but how to measure the weight of the molecules? So the principle is that as you said, we split the molecules in some smaller parts, but let us think that we have molecules with charge, with the charge, thank you very much. And we put such molecules in some place and put it in a strong magnetic field. And if the charges, as the molecules are, or ions are charged, they can move in the magnetic field, and the shape of the trace of how they the route they take, it is different for ions of different weights. And at this place we detect at which position we detect a charge. And based on these positions, we know how was the weight of the ions which were detected here, and according to that we can distinguish of ions of different weights. Okay, so we are able to measure the fragments. And based on the fragments, we can actually guess what was the original molecule. Okay, so based on the masses, it's possible to infer the number of some formulas for the metabolites, or we can also measure the amount of the charge collected here, and from that we can also measure the composition. But one metabolite can be mapped to multiple compounds. So if we have compounds, the original compounds, which were split in order to obtain the charged ions, then we charged fragments, then we obtain. We can only guess what was the original compound. Maybe we have several possibilities. So supervised learning for such experiment would be to map the metabolites to compounds, or for example, if we could infer the chemical formulas, we would like to map them to pathways, signaling pathways in molecules, not molecules, in cells. But I will not go into further details. But in case we have only one space, so here we have two spaces, chemical formulas, pathways. If we have only one space, then we cannot work with functions, but we would like to find out some information about the space. And information can be, for example, to find a structure in the space. What I mean by a structure. Example, analysis of gene expression. The gene expression is usually measured by microarray. I started today, and we can monitor several thousand of genes in one experiment with the microarray. So the activity of thousands of genes, and so we have some measurements. Is it easy to perform microarray experiment? So relatively easy. Is it cheap? Quite. But usually the problem is, in biology, that we have only few samples. And in case of microarray experiments, we have, let's say, 200 samples. But for each sample, we have several tens of thousands values from the microarray. So one observation here is a very long row of several thousand attributes, but altogether we have just tens or hundreds of measurements. So for working with such data, it would be very advantageous if we can reduce the dimensionality of the observation. If we can substitute the original data with very long rows, with matrices, metrics with some small number of columns, short rows. So this is sometimes denoted as construction of supergenes. So instead of having several thousand of measurements, we reduce to some lower number, let's say several hundreds, and then one value in this reduce observation will correspond to activity for many genes. So therefore, supergene. Another task is visualization. We would like to visualize the data. We cannot visualize the result of microarray experiment if we cannot display several thousands of values in some simple picture, which could be comprehensible for human. So the goal is to reduce the data into 2 or 3 dimensions and show it as a 1 or 3 dimensional picture. Another task for unsupervised learning is clustering. Clustering means that we would like to find where are the groups in the data. Why it is important? Maybe if we know that, let me suppose that we have that we measure just one value, and all the values, if I denote them on number line, are positioned at the number line, and if we are able to detect that there is a group of measurements and this is another group of measurements, then maybe this group are similar measurements. So this corresponds to some similarity in the measurements, and we can, knowing that there are two groups is important, and knowing which measurement belong to the group, it's also important, because if we suppose that these are similar, maybe we can split the measured data in two groups and study each group separately with more detail than with working with all of them. For example, if we let's say we say, okay, let us compute the average value. Average value will be somewhere in between, very far from all groups. So this is some value which has no relation to the real data. But if we supply two means for each group, then this mean these values have some meaning, real meaning. Okay, so we will look at three types of tasks. First is density estimation. Density estimation, we would like to know where are the data. For example, if the data are generated from normal distribution, then we can describe the data by by the mean value and standard deviation. Knowing these two parameters gives us some good estimate of the data. So how we can obtain that? We can compute sample mean. This will be the estimate for the expected value, and sample standard variation, standard deviation, not variation, standard deviation, which is estimate for the standard deviation of the random variable. So Gaussian distribution is regarded as the structure of the data. So if we suppose that the data have some structure, we can try to measure the parameters of the structure, and this can be used. So for that, usually we use methods from statistics. This was a very simple case, one-dimensional variable. Of course, we can use a different approach for the density estimation. We can compute histogram. Histogram describes also the distribution of the values, and from the histograms, this will be later on some slide, we can even compute approximation for the density function. So it's very usable. The second type will be data visualization. For data visualization, we need to reduce the number of dimensions in the data to 2 or 3. Even usually not more. Actually, there are methods how to put more dimensions in one picture. Can you imagine such method? I would like to depict some data which are described using such metrics, and if the data have one value, I can just list the values or give a table. If I have two variables, it's important to have the relation between the two values of the attribute 1 and attribute 2. How can I depict it? On a plane, yes. In a plane, I can use, for example, a scatter plot and use the first coordinate as the x coordinate, the second value for the second attribute as the second coordinate, and describe it by, let's say, dot on a plane. If I have 3 attributes, how can I depict 3 attributes? Heat map, yes, color. So either I can try to make 3D plot. It's still comprehensible for humans because we can rotate it and look at that from different angles. But color is can be the third coordinate. So different colors color can express the value of the third attribute. I would like to add more, 4 attributes. How can I do that? Principal component analysis is the method when I reduce the number of dimensions. But I would like to preserve all the dimensions. Excuse me?
Maybe
heat maps? So there are methods how to depict this. For example, instead of just dots, I can use, let's say, a small arrow. Orientation of the arrow can be another maybe even 2 attributes. This is direction, but I can use also the width of the mark. Another, and I have seen long time ago some paper where they claim that they are able to put 11 dimensions in one picture. But of course, this is not so good and not so comprehensible in all cases. So for the data visualization, we will usually suppose that we would like to reduce the number of dimensions to 2 or at most 3. Cluster analysis is what I have already described here, but of course, for the cluster analysis, we can consider variables or data with many attributes. Okay, let us look at the probability density estimation approaches. So the first simplest method is the histogram. We have already seen that in the lab in the morning. So we know we have some data, and we know that the data are from some restricted interval AB, and we divide the interval AB into bins, usually into the same width bins. And for each bin, we count the number of observations which fall within the bin. And the number of observation will be depicted as the height of the bin, and of course the bin can be depicted as one rectangle. Width of the bin corresponds to the width of the rectangle, and height of the rectangle corresponds to the number of observations which fall in the respective bin. So the size of the bin, if we have k bins, is actually b minus a and divided by k, of course, nothing complicated, and the frequency is the number of training data falling into the bin divided by the number of our training data. This is frequency for each bin. So the probability that that point fall within the bin is Ni, and I is the number of data points we have found in the bin I, divided by the number of observations, capital N. my question, how can we approximate the probability density function? From such a histogram. So the idea is, if I have a histogram, I can say, okay, I will approximate the density function as this here 0, constant through the whole bin, constant through the whole bin, constant through the whole bin, and 0 elsewhere. Of course, I should solve what is the value on the border of the borders of the bins. If this function should be approximation for the density function, it must satisfy one condition, which area under the curve must be 1. So area of all these rectangles summed together must be 1. So how can we compute the value for the approximation of the density function? Can we use Ni divided by capital N? No. Why not? If we use this value, Ni divided by N, what will be the area under the curve? Yes.
And the sum of PIs will be 1.
Yes, you are right. But the area of a single bin here is width of the bin times its height. So if we sum all the heights Ni divided by N together, we obtain 1 times the width of the bin. So in order to have the density function, we must divide it by the width of the bin. So it's so simple. And in red, here in the plot, in red is the value for the density function. So I decided that the right border of the bin will be included in the value, will have the same value as inside of the bin, and the left border will not. But this is just cosmetic. Okay. The value for the width is important. We have tried that in the morning in the lab, that by changing the width of the bin or the number of bins, we can change the figure. If the true density is this green function, continuous function, then if we use too narrow bins, so too many bins, then we obtain this histogram, which is not so good approximation for the true density. For the value 800, we obtain something which is quite close, but not completely to the true density. And if we use just 4 bins, it's too rough, and we obtain something which even resembles that the density is a growing function from left to right, which is not true. So this is one problem. So there are many good properties of histograms. So the method is extremely simple. We need to fit a model to the data. So we just take some sample and compute a histogram. And then if you would like to know the density function, we just compute the approximation from the histogram. So it's very simple. The first problem is the width of the bins, or the number of the bins we have seen in the previous slide. So the width can be used as so-called regularizer, which the proper value for the number of bins or width of the bins, it's crucial for obtaining some reasonable information. Maybe we could consider even bins of different width. So we cover the, but it's hard to guess where we should use wider bins and where narrower. If we obtain zero hits in one bin, do we believe that it's really zero? Usually we have no guarantee, because we have some limited sample. So usually we suppose that even if we have zero hits in the bin, we artificially add some small number of bin hits to the bins, to the empty bins, because maybe there is some in the future we could obtain some values in the from the bin. If we have several variables, we consider d variables as the number of attributes. And if we would like to describe the distribution of for multidimensional data, we can use multidimensional histograms. For example, for two dimensions, it's easy to imagine. So we just take some, let's say, square patch and height of the not cube, rectangle, or will be block, or height of the block will be the height of the histogram. We can describe distribution in 2 dimensions or in 3 dimensions. However, if we consider just 10 bins for 10 dimensions, we obtain that we need 10 to the 10th power bins, a huge number. So it's not unusable. And for multidimensional data, it cannot be practically used. When we approximate the density based on the density function based on the histogram, we have discontinuities at the border of the bins. So we obtain some non-continuous approximation. It is possible to do better. Do you know how? You will do that in the first assignment, and the idea is simple. Instead of using bins, we can approximate the values in some interval with some smooth function. For example, something like Gaussian curve, and then we can compute the approximation of the density function as sum of Gaussians. In the trivial case, we define for each observation, we define some small Gaussian, and we sum all the observations, so all the Gaussians from the observation, and we obtain some smooth function. You will do that in the first assignment and use that for constructing classifier. And there is one more disadvantage, at least one more disadvantage, and this is position of the borders of the bins. This example is actually from the documentation of scikit-learn. And we have one-dimensional observations, which are depicted here on the bottom. So at the number line, there are these small crosses, and this is the position of the values. And we have the same sample on the left and on the right. And these two histograms differ just by different borders of the bins. The borders of the bins are on the left. The values, there are 10 bins on the interval, regularly spread on the interval -5, 10. And on the right picture, it's the interval of the same length between -4.25 and 10.75, and also divided into 10 bins. But the picture is very different. So we just moved the boundaries by 0.75. Okay, so these were non-parametric approaches. Let us look at parametric approach. Parametric approach means that we assume that there is some structure which can be described by some parameters, and therefore parametric approach. When we obtain the parameters of the structure, then after constructing the histogram, we can discard the values and use just the parameters. So it's very advantageous because we describe the data by some small number of attributes, and this will describe the distribution of the data. So for example, let us assume that we have a Gaussian data structure. If it is Gaussian, so for the Gaussian normal distribution, what are the parameters describing normal distribution? Mean and variance. These two are enough. So if we have the observations, so our matrix capital X, and we would like to describe the variables using normal distribution. But we have D attributes. So we need D-dimensional normal distribution. And my experience is that our students usually don't understand the multivariate normal distribution. Maybe you know it. I will ask. So what does it mean, multidimensional normal distribution? Formally, I have some formulas there. And in the formula, so if we have one-dimensional Gaussian probability, then we can describe this using such density function. So 1 divided by square root of 2 pi times sigma square. Sigma is the standard deviation. Sigma square is the variance. And times e to the power minus x minus mu. Mu is the expected value squared, divided by 2 times sigma square. But in case of multidimensional normal distribution, the density function is this one. It's 1 divided by the square root 2 pi to d, to the d's power. D is the number of attributes, number of dimensions, times determinant. This value is determinant of covariance matrix, and times e to the power minus, and here we have xi minus mu transposed, because xi, this is one observation, it has d values, it's a vector. Mu is the expected value. Expected value in this case is also a vector. It's expected value for each dimension. Transposed times inverse matrix to the variance matrix times xi minus mu divided by 2. What is our goal? We have some data and we would like to find this multidimensional Gaussian distribution for the data. Okay, what does it mean? We should find expected values. Expected values, yes, we can approximate each attribute. For each attribute, we can compute mean. Simple. Further, we must approximate what we should. So, mean. Next. In one-dimensional case, we need just the standard deviation sigma. But in multidimensional case, we need the covariance matrix. What is covariance matrix? Before giving the definition and how we can work with that, I would like to finish the idea. So the idea is that we would like to find such parameters, the covariance matrix and the expected values or mean, mu, such that if we compute probability, better say likelihood of our observation. So our observation is this big matrix capital X. And the idea is we would like to find such parameters describing the multidimensional normal distribution for which the probability that we observed this that we obtained this n observation is the highest. Maximum likelihood approximation. So this can be approximated as the product of the density function for each observation. So we compute the density function for the first observation, second, and so on. And out of them, product, because we observe all these, we have all these observations together. So the product should be maximal. So actually, mathematically, we just would like to maximize this function, this product, this big product. And the terms of the product are such formula in which we have mu and in which we have the covariance matrix sigma. So just to find the covariance matrix sigma and mu such that this will maximize this product. This is mathematically the formulation. Okay, so what is covariance? Covariance is defined as here the expected value for x minus mu times x minus mu transposed. So simple. It's all. But x is a vector. Vector containing d values. Mu is the expected value for row of the matrix. So it's actually vector of numbers. Mu is simple. We can just compute the sample mean and for each column, and this will be the vector mu. But the problem is the covariance matrix. So the covariance matrix at row I and column J is the covariance between the I's attribute and J's attribute. Mathematically, it's so simple, but we have only the observations. So we must approximate the covariance from our observations. This is just repeated from the slides from the previous part. So this is the notation, and for one-dimensional sum vector, the variance is 1 divided n minus 1 times the sum of square differences between the value of the variable and the mean of the variable. But now we need this for compute the covariance between two attributes. Idea is the covariance should express whether the attributes are independent or dependent. If they are dependent, looking at the formula, what is in the formula? We subtract from the variable its mean and multiply with the other variable, with the difference of the other variable and mean of the other variable. If they, for example, if they grow simultaneously, both variables, so grow that they are higher than their respective mean values, we will have positive difference multiplied and we obtain higher value for the covariance. If one variable is growing and the second is decreasing, then maybe we have obtaining values which are higher for the mean for the first variable and lower than the mean in the second variable. We multiply them and we obtain negative value. So the covariance can be positive or negative, and in which case the variables are independent. If they are independent, so at some point, some observation, they are both growing. In some other observation, they are both decreasing. Or in some observation, one is increasing, second is decreasing, and so on. So if they are completely independent, then the covariance should be 0. If they are going together positively, then the covariance is positive. If one is growing and second decreasing, so if one is higher than the mean and second is lower than its respective mean, then we obtain negative covariance. One more question. The value of the covariance can be positive or negative. Is the value of the covariance limited by some value? Should it be from some limited interval, let's say -1 + 1? No, because here we look at the difference between the value in the attribute and its mean, and such difference can be arbitrarily high. So okay. Now I would like you to write formula for the what is the size of the covariance matrix dimensions? D by D, because these are covariances between pairs of attributes. Okay, and I would like you to say me how to write a formula for one element of the covariance matrix. So we would like to approximate the covariance from our observations, and similarly, as we computed the sample variance, for the sample variance we subtracted the value of the variable minus the mean of the variable computed square and sum all these squares and divide them for the sample variance by n-1 in order to obtain unbiased estimate. And we would like to estimate also the covariance. I have prepared a task for you. I have written some candidate formulas, and I would like you to say me which formula is correct. At the top of the slide, it's repeated the formula. I mean the formula from the previous slide. And now I would like to have it expressed in the respective numbers from the observation matrix capital X. Can you rule out some of the formulas as wrong? Maybe many of the formulas can be ruled out very quickly. So in the formula, mu I is the mean or average for the respective column of the matrix. Mu is a vector, so mu I is ith mean of the ith. Maybe better use j, so mu j is the mean of the j's column. So can you rule out some of the formulas? So we compute covariance between two columns, I and j. Don't look at this I. Now I use I here. So which sum?
Like for example,
A. Yes. Yes, but if we look at the formula A, then K is the variable of the sum, is index the first observation ith column till the dth observation. So we look actually at first d values, not through the whole vector. So it's evidently wrong. So A is wrong. F G H I J. And what about B? Why? So you mean that it's wrong because there is not xi minus mu I times. Actually, it's almost correct. The wrong is only the factor in the front of the sum, because it's divided by n. we know that if we would like to obtain an unbiased estimate for the square difference, then we must divide by n-1. Therefore, we can rule out all the remaining formulas in the first column. What about the right column? J is correct. So J is probably correct. Okay. What about F? Looking to just the first D, observations, it's of course wrong. What about H, I, and J? Besides a typo, here it should be K equal 1, not K minus 1, of course. So this is the answer. Why? I would like to ask, the formula G is strange. Last formula, K equal 1 to capital N, is correct because it's just rewritten from here. Expected value for xi minus mu I. So we take i's. Yes. X. Yes. We take the observation from the case observation, the value of the i's attribute minus its mean, times from the case observation its j's attribute minus its mean. It actually corresponds to this dot product. Why is this dot product? Xi is the column. We subtract the mean and we transpose it into a row, and this will be row times column. So this will be dot product of two vectors. And this is just rewritten the dot product. So it's easy to find that this is correct, and divided by n-1 because we compute the unbiased estimate. This is even simpler. It corresponds to the formula. But why is this correct? It's strange. We should from the last formula we should compute the product x ki minus mu I times x chi j minus mu j. So the explanation is simple, and we must end in 1 minute. So we compute 1 divided by minus 1, and here I can rewrite the sum from the last formula as x ki times x kj minus sum x ki times mu j minus sum of mu I x chi j plus sum of mu I mu j. And let us look at this term. So what is this term? Mu I is a constant, it's one number. So mu I times the sum of x chi I. We can compute the sum of xii. It's actually capital N, the number of rows, times the mean for the ith column. So this can be written as n times mu I times mu j. Similarly, this term can be written as n times mu I mu j. What is this? This is n times mu I mu j. So we have minus n times mu I mu j minus n times mu I mu j plus n times mu I mu j. So we can sum them together and we have minus n times mu I mu j and n times here. So we can put it back in a sum here that we use mu I mu j in all terms, n terms in the sum. And this is the end for today. I hope you understand. If not, please ask. Maybe you can ask at the beginning of the next lecture or during the labs if you have any questions. And next week we will continue unsupervised learning. Thank you for your attention.